Let me show you a tomato variety that I bred. I'm naming it Minnesota Icicle. Tomato breeding is my all-time favorite topic and a hobby that I live for year after year. I'm going to show you how to do this in detail, step by step. But first, just a brief science lesson and anatomy lesson. I'll make chapters so you can skip ahead or rewatch as needed. Here are my thoughts on why you should breed your own plants. One is that you can increase genetic diversity. The more characteristics you have to pick from, the more people have to choose from which they like. For example, taste is very subjective. So some people may like a more mild tomato, other people may enjoy a more acidic tomato. So it's good to have genetic diversity. Also, you can select for characteristics of your choosing. So in my situation, sometimes I do have a specific end goal. In the case of my Minnesota Icicle tomato, I was specifically looking for an elongated paste tomato with that blue or anthocyanin color. And that is what I got with my end goal. But other times when I do crosses, I don't have a specific end goal, and I'm just kind of playing around for fun. So it just depends, you can choose a specific end goal if you would like. Another really important one is that you can select for performance that is adapted to your climate, your specific disease pressures. So this could be insect, pressures, climate, are you in a dry area, a humid area? In my case, I'm in a very short season area. So I'm in zone 4, Minnesota, so a very short season. So my climate is very different than others. So you can, really select down to your specific region. Also, it's, plain and simple, it's fun and exciting. I really enjoy, especially when you're at the F2 stage, that's when you're going to see the most genetic diversity from the parents. You can really start to select for what you want. Also, it's just really cool to play a part in creating something. If you're like me, where you kind of have that creative mind, like you're happy if you're creating something, and I also like to nurture things, so it kind of fulfills that, gives you that satisfaction of playing a part in creating something. It's very fascinating and it's very humbling. Also, let's not forget that you do get to eat your mistakes. It is a little cliche, but it's true. I have to say, I've never really bred a tomato that was so awful that I couldn't eat it. But some of them maybe just aren't superior enough that I want to proceed with them. But regardless of what you think of your results, you get to eat it, and that's fun too. I want to talk about outbreeding plants versus inbreeding plants. I'm standing in front of my winter squash right now because this is an example of an outbreeding plant. So outbreeding plants are considered monoecious, meaning that there's a male blossom and a female blossom separate. So the male and female parts are separate on the plant, separate blossoms. I'm going to zoom in and show you a close-up of that so you know what I mean. So let's zoom in and look at this male one. Now it is a little late in the season because it's kind of the end here where they're not starting fruit anymore, but this blossom here is a male. Yeah, there's some bugs in there right now. So that would be producing the pollen. So this whole blossom here is just a male. There's no female part here. So outbreeding plants rely on insects and or wind to do the pollinating because we need to get the pollen from the male to the female blossom. So bumblebees, honeybees are good examples of that, as is wind in an example of corn per se. So now I'm going to show you what a female blossom looks like for comparison. So this would be an example of a female blossom. Now it's already been fertilized, so it was already opened, fertilized, and is producing a fruit here. But just to show you an example, if you tear the blossom open, that would be a female blossom that's already been pollinated. So this is a female blossom, and the male blossom is separate. And you can see right here, there's a bumblebee right now pollinating on a tomato blossom. So those are examples of outbreeding plants. On the other hand, if you turn over to this side, you're going to see all my tomato rows. And the tomato blossom is an example of an inbreeding plant. So that means that the male and female parts of the plant are all within the same blossom. So they inbreed or they self-pollinate themselves. You can, expedite or ensure the pollination by use of insects. So bees will come along, as we just saw, and pollinate the blossom and kind of vibrate it. You can also use an electric toothbrush and go around to kind of make sure that pollination happens, but it's not necessary. All the parts are there to pollinate itself. Let's review some genetic basics. An allele is any of the alternative forms of a gene that may occur at a given locus. Homozygous refers to having identical alleles for a single trait. Heterozygous is having two different alleles. The genotype is the chemical composition of the DNA, and the phenotype is the observable traits of an organism, like brown eyes versus blue eyes. Using my Minnesota icicle tomato as an example, I crossed a beefsteak tomato with an elongated tomato, and I got various phenotypes of tomatoes. In this video, I am referring to traditional or classical plant breeding, which are field methods involving whole plants. This is not GMO and does not involve transferring genes from an organism to an unrelated one. Classical plant breeding transfers genes through normal reproductive processes. To quote one of my favorite books, which I will reference, "It is merely a speeding up and guiding of natural reproductive processes." Do you remember Mendel's Punnett square from school? Well, we're going to look at that today using my Minnesota icicle as an example. Gregor Mendel was an Austrian monk who performed many crosses with pea plants in his monastery garden. He was able to discover how characteristics are passed down to each generation. This is where we get our Mendel's laws of inheritance. Now we know that the field of genetics is much more complex, but we had to start somewhere. There's a lot we know and a lot we don't know, and this is why you and I should keep growing and keep learning. In this example, we are crossing two heterozygous tomatoes, where a capital R is for a round tomato, which is dominant, and the lowercase r represents a long tomato, which is recessive. In the upper left corner, we have homozygous dominant, so that phenotype will be round tomato. The two squares with capital R and lowercase r are heterozygous dominant, so that phenotype will also display as a round tomato. The lower right square is homozygous recessive, which will display a long tomato phenotype. So for this example, 25% of the time you will get a long tomato and 75% of the time a round tomato. Now let's switch to crossing a heterozygous dominant round tomato with a homozygous recessive long tomato. In this example, 50% will display the round phenotype and 50% will display the long phenotype. Although Mendel gave us a great starting point, not all alleles behave so clear-cut. Alleles aren't always fully dominant or recessive, but may instead display codominance or incomplete dominance. Incomplete dominance is when two alleles produce intermediate phenotypes. Codominance is when two alleles simultaneously are expressed. My purpose for this video is to show you how to do the crossing and to encourage you to give it a try, so I don't want to dive too deep into the bookworm aspect of it. I will reference a couple of books for those of you that do want to dive deeper into the topic. When I made this cross about 10 years ago, it was much less than 25% for achieving that elongated phenotype. If I hadn't grown out hundreds of plants, I may never have achieved this end goal. Not only did I have round, but I also had heart-shaped as well as various elongated shapes. So, if your spouse asks you, do you really need to grow that many tomato plants? You can reply, why yes, dear, this is necessary in the name of science. I want to talk about the process of the breeding. So you want to select the mother and father plant. So the plants that you select should be open pollinated or stable. In other words, homozygous. So you're going to pick the mother and the father, make that cross, and then that fruit, you'll save the seed from it. And that seed, when you grow it out next year, is considered the F1. So the F1 is what you would consider a hybrid. When you buy a hybrid variety at the nursery, for example, that's what that is. It's been crossed, and then the next year you grow it out, that's a hybrid. You've heard of hybrid vigor, so a lot of times hybrid will be very healthy plants. So you do not need to grow out that many of the F1s because they're all going to be uniform. So just a few is fine, just enough to get, adequate seeds. Then you're going to self-pollinate or inbreed that again. So the following year, you're going to have an F2. So the F2 is one of my favorite stages, and this is where you're going to want to grow lots of them. You're going to have the most genetic variety to select from at this stage. So you're going to see, a lot of traits from the mother, a lot of traits from the father, and a whole bunch of variety of combinations. So this is where you have that opportunity to start selecting for what you want. So grow lots of them and have fun with it at the F2 year. Then you want to keep continuing to, self-pollinate and select each and every year. Once you get to about the F6 to F8, it's considered to be more stable. You're going to start seeing less variation and have a more stable variety at this point. So each generation will display less and less variation. Once you get to the F8, I hope that you share your varieties or sell them and let other people enjoy your hard work. Next, we get to learn tomato anatomy. This is absolutely essential, much like a healthcare provider would need to learn human anatomy before performing a procedure. Honestly, I just love the beauty of these plants. I could just look at this all day long. Starting externally and working inward, we have the sepal, which is open in this photograph, followed by the petal, which is partially open. Again, we're looking at an opened petal, and in the center there is an anther cone. The anther cone is the male part of the flower where the pollen is produced. Tomatoes are considered perfect flowers where both the male and female parts are within the same blossom. With the anther cone removed, now we can see the female anatomy of the blossom. At the base is the ovary. When fertilized, this will become the fruit. The stalk-like structure protruding from the ovary is called the style. At the tip of the style is the stigma. The stigma is the part that will be receptive to receiving the pollen. When the cross is successful, the ovary will swell into the fruit, and you can see the exposed stigma. Another example of a successful cross with the stigma still intact. These are the materials that I recommend and use myself when breeding tomatoes. The first on my list is an electric toothbrush. Make sure it's fully charged before you go out into the field. We'll be using this to vibrate the blossom to achieve pollen from the anther cone. Toothpicks, a small scissors, a dark lens or dark colored sunglass, hot pink marking tape, and a permanent marker. I'll meet you out in the field and we'll get started doing the breeding. Step 1 for breeding is we need to select the proper male and female donors that are at the proper stage of maturity. To select the male donor, both the sepals and petals should be retracted back fully. The anther cone is exposed. This is the ideal time to collect pollen. If you are too early or too late, you may not get pollen. To select the female, I'm going to show you several examples, as this is very crucial in determining the success or failure of your cross. Select a blossom where the sepals have retracted back, but the petals have not. These are all examples of the correct timing. You'll notice that the sepals are retracted back, but the petals are still in a closed or cone shape. The selection of the female is very important. If you were to select a blossom that is too immature, you risk that the stigma may not be receptive to the pollen. On the flip side, if you select a blossom that is too mature, it has likely already been pollinated by the anther cone. Likewise, I'm going to show you examples of what not to select to help you differentiate. In this example, it is too immature or too early. As you look closely, you can see that the sepals are not retracted. This would not be a candidate. And another example where the sepals are not retracted. This is simply too immature. To illustrate too mature, you can see here that the petals have opened. And in this example, the petals have completely retracted back. These examples indicate that fertilization has already occurred and the petals are now withered. This is too late. This blossom on the left is the correct female timing. The blossom on the right would be incorrect because the petals have begun to open. To summarize, this is the correct selection for the male, and on the right, this is the correct selection for the female. I will meet you out in the field and let's get busy starting to make the cross. Step 1 is emasculation, and it's exactly what the word sounds like. We are going to remove the male parts of the blossom. I have selected the female parent. As you can see, the sepals are open, but the petals are not. Using my toothpick, I'm going to retract those sepals so that they are out of the way, and I'm also retracting back the petals so that they are also out of the way. We need to remove the anther cone. I will be using the tip of this toothpick, and I'm carefully getting to the base of the anther cone to kind of lift it up and away. Some breeders may choose to use a tweezers instead of a toothpick to remove the anther cone. This is just how I like to do it. There's certainly more than one way to get to your goal. You can see that the anther cone is getting lifted away piece by piece. Take your time. We do not want to damage the style. If you accidentally damage the style, you would have to find a new blossom and start over. Once the anther cone is removed, now you can clearly see the style and stigma. Next, I'm going to take my scissors and trim away the petals and sepals. Be very careful when you're doing this not to bump the style or stigma. We don't want to damage the female parts. And now we have a fully emasculated tomato blossom. Let's look at one additional example of emasculation. It's such a delicate and detailed procedure that I think it's important to see it more than once. So again, with my toothpick, I am retracting back the sepals and petals. This will expose the anther cone. The anther cone is going to be more firm compared to the petals, so I find that it's helpful to just kind of pick at one piece to get it started. And once it's started, it's easier to get the remainder of the anther cone off. In other words, once you have an opening, I find it easier to get to the base of it to remove the remainder of the anther cone. So right here, one piece came off, and now you can see how much easier it is to get the remaining pieces of anther cone. It just slips right off. Grab your scissors and trim those petals and sepals. The reason I trim is to make the next step easier and for identification purposes later on. And here is the finished emasculated blossom. Let's look at some still shots of emasculated blossoms without the wind blowing. So these are emasculated without being trimmed. You can see that the sepals and petals are still attached. These are examples of fully emasculated blossoms that have been trimmed. If you're a visual learner like me, it's really nice to see examples of all the different varieties of tomatoes. Each blossom is different. Beefsteaks are very different from cherries, for example. I never get tired of looking at garden photography. And for the final example, a very nice close-up. Next, we need to collect the pollen. So choose the male candidate, such as this one or this one. Grab your sunglasses and your electric toothbrush. I like a dark lens because it's easier to see the pollen. I'm using an electric toothbrush with the head removed. We need this vibration to extract the pollen. Position the lens underneath the anther cone. When applying vibration to the anther cone, you can clearly and visibly see the pollen on the dark lens. This is an adequate amount to pollinate one female. If it's not enough, you can always select an additional male of the same variety. An additional trick that I've learned is you get better results when you place the toothbrush on the tip of the anther cone. Here I'm doing it at the base and I'm not getting quite as much versus doing it the other way. Here's what the pollen looks like up close on the lens. The next step is applying the pollen to the stigma. Bring your chosen pollen over to the emasculated blossom. Gently rub the stigma along the grains of pollen. The key is to be gentle and not break the style. Example number 2, I have a real nice amount of pollen here. So I only apply pollen once. I do not go back and apply pollen another day. And I find that I have probably about a 95% success rate. In my experience, as long as you select the male and female at the proper stage, you're good to go. Next, I trim off all of the remaining blossoms. Unlike squash, which I would pollinate in the early morning hours, I tend to do the tomato crossing in the midday. Cherry tomatoes will have a very thin and delicate style, whereas beefsteak tomatoes will have a stocky and bigger style. Again, I'm going back to trim off those remaining blossoms. It makes it easier to identify, and I want all of the energy going to that crossed fruit. I'm sure by now that you can tell this is a topic I dearly love. Here are some detailed photos of pollen on the stigma. Absolutely beautiful. Now it's time to label your cross. This is the second most important thing, the first being selecting the male and female at the proper maturity. I write down who the male and female parents are, and I assign it a number. In this cross, I'm using Mila as one of the parents because it makes an excellent sauce that's very thick, but I'd like to make some improvements to the plant. With a permanent marker, I will be writing down the assigned number on some hot pink Surveyor's tape. I like the hot pink because it's much easier to identify in a sea of green foliage. An optional step is to cover the exposed stigma to prevent pollination from insects. I say that this is optional because I do not do this step. Although it is possible for a honeybee to come and land on this stigma with pollen on its legs, it is also unlikely because the bee will not be as attracted to this blossom. Remember, I have removed all of the yellow petals. Nonetheless, if you are concerned about this, you may choose to wrap the blossom in some kind of wrapper, such as a nylon, tulle, cheesecloth, something breathable. Make sure to not tie your label too tight. Next, how to identify a successful cross. I only have one example of an unsuccessful cross. You can see that the stigma is gone, everything is pretty much desiccated, and there is no fruit. A successful cross will have the ovary swollen into an immature fruit. The style and stigma are visible, and the sepals that have been trimmed are also easy to identify. How exciting it is to see your successful work. And a harvested cherry as the mother plant. After you've done your crosses and the fruit has ripened, you need to locate them. There's 3 things that I do to help myself locate these crosses. One is I use that hot pink marking tape. It really makes it easier to see when you're looking in a jungle or a sea of green plants. Once you've located the marking tape, and we're going to zoom in right now, you're going to notice that we had trimmed the sepals of the plant. So this one here is ready. I see the hot pink marking tape. If I follow that down to the fruit, you can see that the sepals, these green things here, have been trimmed. So remember, we trimmed those with the scissors. So now, that's easy, one way of identifying it as well. Also, all of the neighboring fruit has been trimmed off. So it's a single fruit. There would have been tomatoes here, here, and here, but that's been trimmed off. So it's pretty obvious here that this is the one. So the hot pink tape, the trimmed sepals, and the fruit, missing fruit on that branch. You want to make sure that it's ripe. Now this one is ripe, so I'm going to go ahead and pick it. So we want to make sure that we don't mix up this fruit with a different one, obviously. The hot pink label is going to show what I number my breeding plants, so I can look up and see what the parents were of this cross. So the next thing we need to do is get the fruit out of here so that we can grow this again next year. I have a really detailed full video on how to do tomato seed saving, so I will have a link in the description so you can get instructions on how to do that. A quick distraction from my garden companion. Lucy, are you enjoying the shade of the tomato plants? Are you enjoying that shade on the hot sunny day? She also likes to lay under the zucchini and under the big rhubarb leaves, don't ya? Yep, she likes that. She likes to hang out in the garden. I have a really good example of some F2s that I want to show you. So the F2 is where you see the most variability from both the mother and father coming out. And this is my favorite stage of doing the breeding because you can really select for different traits. This is a great example though, showing how you can get vastly different results from the same cross. So go ahead and look at this closely. The first one we have is kind of an elongated egg shape. And it's a nice red color with kind of some orange mottling to it. Kind of some faint stripes, kind of mottled look. It's very attractive. And I thought that I would probably be selecting for this one based on looks. Versus the same line, we have kind of a squat pear-shaped one that's a real faint yellow color. Again, this is the same cross, but vastly different results. And what really surprised me is that I really like the flavor of this yellow one, which is shocking because I do prefer stronger flavors like acidic or tangy, etc. And usually the white or yellow tomatoes are kind of more mild, so this surprised me how much I liked it, and therefore I'm really excited to carry this one forward into the F3. But just a great example to illustrate here, same cross, different results. I hope that this video has inspired you to try breeding your own tomatoes. If nothing else, I hope that you found it interesting and educational. If you'd like to learn more, I have 2 book recommendations. The first one is Plant Breeding for the Home Gardener, and this is a nice light read. If you'd like to delve more into the science of it, I really recommend Breed Your Own Vegetable Varieties. I'll have links for both of these books in the video description. I really hope that you're able to make your own variety, name it, and share it with others. If you have any resources for me, such as good book recommendations or social networks, I would love to hear from you. Please leave a comment.